All right, thank you. All right. Okay. Let me say a few words here first, and then, yes. So thank you all for coming. I know it wasn't necessarily easy to get in for some of you today, and I greatly appreciate all of the in-person attendance. I know we also have some attendees on Zoom. The way I think we're going to use the Zoom is if you have questions, please type them into the chat, and I will read them at the end. But that said, if there are people here in person, we may get to those questions first. But by all means, please do engage with the chat on Zoom if that's the way you're attending today's talk. Okay, so it's my great pleasure today to reintroduce Bingshu Wang. Bingshu grew up in China, and she got her bachelor's degree in physics at Peking University. While she was there, though, all of her research projects were actually in the areas related to neuroscience, both sort of human behavior and also her introduction to the macaque visual system. Upon graduation, she applied to neuroscience PhD programs, including ours, and we made the good decision to admit her. And we were very fortunate that she decided to come here to do her PhD. At WashU, she joined the lab of Carlos Ponce. And there, she and Carlos collaborated over several years on a whole series of very high-impact studies that really sort of lay at the boundary between, areas of neuroscience and areas of AI. So the sort of main theme of this research was to try to use the, then nascent technology of image generative AI as a way of synthesizing images that could drive neural activity in some of the higher visual processing areas, and sort of try to optimize and tweak those stimuli to maximally drive those neurons. So this work was really interesting because it was really exploring this brand new sort of computational technology. And it both, I think, the papers ranged from, sort of ones that were really focused almost exclusively on characterizing responses in the macaque visual system to ones that were really looking at just what are the basic properties of the image generators. And that was necessary because this is, exploring visual space is a very difficult thing to do. It's so high dimensional. And so to do so effectively required leveraging both insights about the structure of how these image generators actually performed, together with an understanding of what was feasible experimentally in neuroscience. And so I think there are relatively few people who could have pulled that off, but Bingshu really did a masterful job of that work. When she defended her thesis, she joined a new institute at Harvard, the Kempner Institute for the Study of Natural and Artificial Intelligence. This got started just in 2022, and it's a really exciting sort of training ground for people like Bingshu who really sort of live at this interface between the world of AI and the world of neuroscience. So I think we'll hear at least about a subset of some of these activities from the seminar today. So please join me in welcoming Bingshu.
Thanks, Tim, very much for the very kind introductions. And basically, it has alluded to many parts of the talk, and hope I can deliver a good job of all these contents. And thanks, everyone. It's a very fortunate to come back to this stage and talk about many of my works. I mean, many of those started here. So to start with, some of the most remarkable aspects of human intelligence is in art and scientific creativity. And some of my favorite pieces of them is like the Starry Nights in Van Gogh or the Einstein field equations, which are things that nobody has seen in their sensory space, but the authors of them can create them somehow from their imagination and creativity and ends up this kind of time-lasting pieces. And that has driven somehow my research. And I was curious of this kind of creativity in humans. And one piece of advancement in AI that links to creativity is this thing called generative models. Just to briefly introduce what they are. So generative models, they basically learn from a set of samples, like a set of X, and they learn a distribution under them. And then they can draw new samples from the distributions, and voila, that's it. So there are many classical ways of doing so. For example, fitting a Gaussian to your data and draw new samples from Gaussian. That's the easy way to do so. But in the last 10 or 15 years, there has been significant developments basically due to the advancements of deep neural networks. So we call this like deep generative models. From the early days, there has been like generative adversarial networks like GANs. And these are some names I'm going to mention today, big GANs and deep sims, which are really relevant to our work. And more recently, many of you may have noticed works like Stable Diffusions or Soros or Movie Gens. Those are all based on diffusion models, which are more related to my recent works, and I will mention them in the end. So these are some examples of their creativity. For example, they can create images that nobody has seen before or faces that doesn't exist in this world. And they can even interpret prompts and create things that combine features that doesn't occur in nature. So in my past work, I have spent some time trying to scientifically understand these generative models about GANs latent space structures, or how does diffusions create samples, or learning the data distributions. And I will skip most of those since the audience is neuroscientific, but I'm happy to dive into those if you're interested. But based on this understanding, I'm trying to leveraging these tools for generative AI for neuroscientists, both as like tools and also as conceptual frameworks, perhaps. And in the end, hopefully, we can bring something back from neuroscience to the AI world. For example, studying the generative systems as model organisms. And let's dive into one of the examples I'm trying to do this in my past work. So the first part is driven by this question of how should we study the visual neural code in ways that surpasses like human imaginations. The problem of neural code is defined as such. So our visual system, so this is a diagram for visual system primates, the ventral stream especially. So it receives images from a natural image space, like my cats, and it processes through the stages of the visual cortices and transforms them into spikes of neurons. And one way to understand those is as a vector in the vector space. So we call this the neural population response space. And the interesting thing is the mappings from image space to like the neuronal response space. This mapping, we call it F. So one seminal question for this line of work is how the neural code is transformed through the visual cortical hierarchy based on this diagram of Vanessa and Feldman. And we are going to especially study 3 stages from the early one, the V1, V2, and the V4 and posterior IT in my talk. So to study this problem, the classic approach to study visual neural code is to start from some sets of design stimuli, like this one looks like a Pac-Man's ghost, and then you record neuronal responses of it, which is this PSDH, or Paris stimuli time histograms. And seems the neuron likes it, or in the sense, like responds highly to this image. So the creators of the image can morph it and generate different variants of the image and see if the neuron likes it anymore. Since the neuron doesn't interest in this variance, and maybe after some trial and error, you will convert to something that's highly activating to the neurons, but also minimal in the sense of features. And this is like the classic works of Keiji Kanaka from like 30 years ago. So this is good, but there are also certain limitations of the classic approach. For example, the natural image space has been immense. For even a low resolution image like 64 pixels, there are so many combinations of images that you will never get hope for exhaustive search, even in the neighborhood. Further, people have found that some neurons are responsive to features that are rare or even non-trivial in the visual sense. For example, this classic work of Jennifer Aniston cells, and what if I have never watched this TV series or know this person, then I have no hope of generating such images for stimulating such neuron. So in a sense, us experimentalists are also limited by our own imaginations of what the stimuli space is. One thing we have not leveraged is like natural images exhibits the low dimensional structures. So the images does not just stay around in like random places, but they have underlying low dimensional structures among them. What I mean by that is like random combinations of pixels doesn't give you a natural looking image, only some special combinations is. And so they follow a certain image distributions. And this sense is kind of vague until we have some sort of generative model that instantiates the image manifold. So from 2014, we have the generative adversarial networks, which maps vectors into images like such. And when the vectors continuously change in the vector space, the image will also continuously morph in the image space. And you can think about these models as smoothly parameterizing image manifolds. What this allows us to do is to generate and synthesize natural variations along the image manifold. So if you just sample or varying your latent points in your latent space, you will sample some plausible variations of your seed image on the image manifolds. And that's the trick we leverage for developing our approach of neuron-driven sampling. So you can think about it like a more modern way or AI-based way to do the same study of KG Kanaka. We start with like a seed image like this, and then we sample variants of it on the image manifolds through the generative models, and then generates the next batch based on the neuronal responses simulating the experimentalist mind. In some sense, this is not too different from what we do when we play with Instagram or Facebook. It's basically using our responses of the neurons, our feedbacks, to update the images for the neurons. So we can call it also like Instagram algorithms for neurons. This line of thought has been there for around 20 years. And recently, it has been rediscovered from several labs, including ours, and DiCarlo lab and Andreas Tolia's labs in many places, that we can use neuronal feedback to sample the images and design experimentalists in more automated ways. And we are on this branch. So how does it work in concrete terms? In this experiment, we'll have a macaque monkey sitting in front of the screens, fixating on the center of the image. And we will have a bunch of latent codes, which will be rendered through the generator into images, which looks fuzzy. And they will be sent back onto the screens and to show in sequence to the monkeys. And the electrode in monkey's brain will record like PSDHs from the neurons, which will be sorted and feedback through an objective function, usually like the average firing rate of that neurons. Then it will come back to the optimizers, which will tell how should we change the latent codes in the latent space. One of my contributions to this line of work is to build efficient closed-loop controllers for neuronal activities. The previous approach developed by my advisor and their colleagues is using genetic algorithms, which basically treats like the latent codes like a gene and mutates points on this gene. And I leveraged some geometric structure in the space and build a vector space addition algorithms, and which in fact like makes the algorithm faster and achieve higher activations. In follow-up works, we also leverage other geometric structures like spherical geometry or Riemannian geometry in the space to build better algorithms, which I will skip for this point. But in the end, the algorithm will work like such. So for each generation, you will sample a bunch of points in the Gaussians, and you will evaluate these points by sending it to the generator and let the monkey looking at those. And then you do a weighted average of the top samples, which are the ones the neuron like the best, and create your next batch of Gaussians based on the new means and the new covariance. And in the end, we find the covariance is kind of isotropic, so it's like basically identity or sphere. So how does it look like when you really do this experiment? So this is one session in vivo recorded from the monkey IT neurons. And top here, you will see the best images in each block. In the second one, you will see the mean firing rate across blocks. And down here, you will see like the mean PSDH of each block. And this is how it looks when the experiment runs. Each frame is like one block in this session, and in each block, like there's like around 40 images showed in sequence, and we are just visualizing the best image and also the mean PSDH. As you can see, the PSDHs keep going up and converging to something that seems high. and the image will converge from something that is like colorless textures to some object that seems object-like in some sense. Part of the image will flick back and forth and varying, and seems the neuron doesn't care as much about those parts. And some part of the image seems stay put, and seems those parts are crucial to neurons activations staying high. So simply put, what have you learned from this kind of experiments? There are at least threefold things we can learn. One is like we can estimate the true dynamic range of neurons better. Previously, by sparse sampling images, we are hardly reaching their max in some sense. And we can also estimate the contents of the neurons in the sense like maximally activating images. And most interestingly, the meta fact that we can steer the latent codes via simple optimizers through the neurons is interesting. Because if you think about it, the neurons are very nonlinear functions of images, and also the generators are nonlinear function of latent codes. How could these two very nonlinear things work together and get something so efficient optimizing through it? So that's a meta question I will try to answer toward the end of the talk. I think at this stage, you may have many questions in your mind. So I listed some frequently asked questions, and we can discuss them like towards the end. And these are some quick answers I can give. So firstly, are they stable across runs? Because it's maybe a non-convex optimization problem, so likely not. What we find is that neurons will converge to different points in the space, but have similar visual features. And also, like, how do you interpret the results? I will answer that in the next part. And how easy it is, it's very robust, so it's not very hard. Finally, like, how about neuron population codes? I mean, we will gear towards our in the future work, as you can see. So one thing you may notice that even for neurons, for this posterior IT neurons, the image looks very complex. So it's not like a single thing and like more like abstract art from Van Gogh. So you may be tempted to interpret it with using our intuitions. Maybe this thing looks like a face of a dog or a duck, so maybe that's a face neuron. And that one looks like an ear of a monkey, so maybe the monkey ear neuron. And that's one classic way people doing things, which is using our intuition to decipher images and see what are the neurons are about. And that's also a problem for like natural images. Like if a neuron is responding to like a dog image, is this a dog neuron? So we try to do better by inferring the critical part through like computational or predictive modeling of the neurons, which takes human kind of out of the loop. One classic way for modeling neurons is through this linear, nonlinear models. We have some deep visual features from a pre-trained neural networks, and also some linear ways combining these features. And there's a nonlinear function. One innovation we made for this line of work is introducing the so-called wear and walk factorizations. And there's part of the way it's just about the spatial mask. So localizing which parts of the image the neuron is caring about. And this part of the way it's about features, which tells us what kind of things neurons are caring about. And there's a free parameter of number of parts, which can be identified through cross-validations. If we happen to fit these models, it may tell us how to predict the neuronal responses and attribute important parts of the neurons, and even simplify these prototypes through re-examinations of the model. So we find that our model is doing better than localizing the spatial dependencies of the neurons. Many classical approach will under or overestimate the neuron's receptive field or dependency on the pixels, while the factorized regression method will localize much better, corresponding to the ground truth of the data. Based on this fact, we are more confident in using the model to interpret the neurons. So this is the same neuron as you see in the example sessions, and the model can predict the neuron's response in held-out data robustly, and our model can tell us which are the parts the neuron care out of the both. And these are the three parts. One is the central object thing we saw, And these two are maybe the background or surround of the object. And the relative weights can be seen in this composition mass. So the most important thing is indeed like the central object. And you can even re-evolve your model and get some images that is simpler than the original one. In some sense, denoise the original max activating images. For more classical neurons like V1 neurons, this is evolved images. It does not look very clear, but through the factorized modeling, you will see there's a central part corresponding to like Gabor-like features and some other surrounds corresponding to like texture or backgrounds of that Gabor. These are more examples for the three areas we are recording from V1, V4, and posterior IT. One interesting thing we find is if we cross-validate and determine the optimal factor numbers or how many parts the neuron needs to fit their neuron activity, we find V1 really needs only 2 parts, V4 needs like 3 and 4 parts, and IT needs 5 or 6 parts. So the exact number will vary from model to model, but the trends of needing more and more complex parts progress from V1 to IT. So that concludes the first part of the talk. So neurons can efficiently guide image optimizations using our algorithms and from leading to prototypes or max activating images. And our factorized modeling approach can localize important parts for the neurons. And we find the complexity of the tuning of the neurons will increase along the visual hierarchy. One thing I think is interesting is we just think neurons, we call this generative approach, not just because we use generative models, but maybe we can think about neurons as generative models themselves. So this is a line of deep generative model called energy-based models. If we think about neurons tunings as the energy landscape, then our evolutionary process is somehow like this dots in this scene, which is climbing the mountains and reaching the peaks. So in some sense, we are using Landryman-like dynamics to sample from the distribution or peaks of these neurons. And that may be one interesting interpretation of our experiments. And that's the first part. Is there some questions? I can, oh, please. So why do you think they're single peak? I think they are not. Yeah. So after re-evolutions or repeating these things, then you will see multiple converging to multiple peaks or multiple images, but they will share some interesting similar features. I can show you one example of this. Like, so actually, this is showing like the peaks we find over like 8 months of the same channel on the array. So you can see there are some differences of the exact image or exact prototypes. And so that's kind of talking to there's really multiple peaks, but they also has some shared features like the dot and the white background. So I think that's my interpretation. Could there be something much good? Yeah, we do. So there are some interpretation-ish problems. So mostly we find neurons like such, like they're stable. And in cases when they have converged to different things, usually we conclude that the electrode has shifted in some sense. Like, so it's kind of hard to do it. Yeah. Mm-hmm.
You establish the number of important components by looking where predictability was maximized. Yes. It seems that that's an assumption that maximum predictability is really what says what the neuron is coding. Can you sort of defend that? I mean, is it so an obvious case would be a neuron that is really intending to generate a random response. But I'm thinking there might be other cases where you could make an argument that predictability, that your ability to predict is not the best measure of what the neuron's really doing.
Yeah, I think it's a quite a deep and meta question. So I think that's an assumption basically based on, I mean, that through all the works of like trying to understand visual code, basically kind of we understand this function, like at least at this part as like a static function of the image. And if it's not due to the facts like adaptations or those kind of things, which indeed has a small role in these things, then this approach will fail. But most of the work, like modeling neurons, you kind of need to think about them like some functions. And there may be more general framework, like think about them like generative models themselves, like emitting a distribution of responses in some things to signify some distribution. But here we are not using that. And I think that's a kind of common assumption people make to find good models for neurons. Yeah.
So I have a question, and if you feel the rest of the talk addresses it, or if you want to save it for the end, that's fine. You don't have to answer now. But so my question is this: so I find these results, pretty cool on one hand. On the other hand, I'm always a little puzzled because I feel that at the beginning the question was, how does the visual system, process, decodes, whatever, treats, let's say, natural images, and at the end of this, I feel that the answer that was the question that was answered was, how do we drive the neurons best? But that's not the same thing.
Yeah.
So, I mean, how do you go from where you are, from these results, back to what I think is the real question, which is, how does the visual system treat visual images? That link to me is unclear. I don't know if you want to say something about this. And again, if you want to say it later, that's fine.
Yeah, I think towards the end of the talk, I may tie this back to some more conceptual advances along those lines. But indeed, at this stage, we are basically showing you results for understanding single neuron codings and how their responses over the image space like spans or the geometry of that tunings. But how the whole image, how the whole visual system works regarding the whole image space is like a broader questions like relating to population code and those kind of things, which I haven't shown you. Yeah, hope I can answer better after we present the final parts of the talk. Yeah.
Any other burning questions? Well, let's do one more and then we'll let you move on.
So you compared the images with art, right? And it seems that all the images are indeed subtypes of a specific style of art, which suggests that maybe the generator doesn't explore the full possible space of all images that are possible. Any comments?
Yeah, that's a great point. And in the third part of the talk, we are exploring different types of generators and different styles. And that's exactly the question that drives us. Yeah, thanks for mentioning that. Oh, those are other examples for the re-evolution from the same things over time. And it's kind of stable, but not the same image. So going back to the talk. So one thing I learned from physics is like geometry is important, and that's one thing I like. So I'm trying to think about some geometric problem in this closed loop system. So we have a parameter space, which is our latent vectors, which are mapped to images through the natural image manifolds. Then the system will look at the images and generates responses in the neural activation manifold. So there are two types of geometry or mapping that are interesting. One type is the geometry of the generative model, which is the mapping from latent to image, and one that is the geometry of neural tuning on this latent space, which we are leveraging. So in the second part, I'm going to tell you a little bit about how the geometry of the image manifolds in the generative model forms and how they're relevant to our neuroscientists. It's driven by this question, how can we understand this latent space of GANs? So why that's a problem? For a long time, we know like the latent codes, or Z, are sampled from the Gaussian distributions. So in some sense, it's an isotropic Gaussian and with white noise. So it's a spherical thing. And if you do PCA on it, it will just tell you random noise. So how should we interpret such space without like a reference frame or fundamental like framework? So I try to tackle this problem through this geometric framework by thinking about generative models as space warping. So you map one vector to images, and the distance in the latent space, in some sense, is like the latent distance or Euclidean distance. But what really matters to us or to neurons in the visual brains is the image distance on the image manifolds. So in a sense, you can define the distance in the latent space by pulling back the perceptual distance in this image space. And this could be approximated by deep neural networks that approximate our perceptions. So thinking about this, it's tempting to analog this to another problem, which is navigating a warp space like space-time. So imagine yourself in the latent space and walk around, and how you change the image is not very intuitive. And there's some directions that may take you far away and some directions that seems make you stay put. So neurons are capable of leading the optimizer to walk in such space. So how does that happen? So we try to solve that problem through the framework of Riemannian geometry, which is also the framework for understanding space-time in general relativity. So to give you like the one-on-one or brief pitch of this framework, so we have at each point a metric tensor or symmetric matrices, and basically describe how locally things are distorted. So this local mapping will map a square to something maybe like this, and some directions will have a higher amplification and some directions will be diminished. And we can use this matrix to understand the local norms of the vector or length of it or angles of it. Most importantly, if you perform the eigen decomposition of this matrix, you will get the orthogonal basis, which has orthogonal effect on the manifold. And so this is like the classic basis we are going to use for describing the mappings. One technical contribution made to this line of work is to compute the Riemannian metric tensors through autogradient techniques in machine learning. So using these techniques by double differentiating the distance function, we'll get the Hessian matrix or the distance, which we find is mathematically equivalent to the Riemannian metric tensors. And it will give us this local measurement of the distance structure on the manifold. Our code is open sourced. So one way to look at it is how does the directions on the manifold means? For example, if you center around a single point, what are the different directions? So we started with this one image of the dog in a big GANs noise space. What we will find is like different directions will signify highly different or anisotropic meanings. Some directions basically lead you to stay put. There's no significant change, and some directions will lead to very prominent change in the poses and maybe the background of the dog. Similar for other generative models. So we can conclude that the neuron in this space, the rate of image changes dramatically across the different eigenvectors or the spectrum. And we show this also in our GANs we have used for our previous work, which is the DeepSim or FC6 GANs. So it has a lot of small eigenvalues, around like 3,000. And in some sense, this space is a non-space. And we also find that for other prominent GANs in the literature. What we mean by non-space is the following. So if you try to traveling in the latent space and even travel a long distance, the image will stay put. So this direction is basically nullified or ignored by the generators. So that tells us there's no need to search in such space because that's not informative for the visual neurons. And the following question will be, if we find such a non-space or such a prominent geometric structures, is this local or global? If it's a global, then it's much easier for us to just reduce the space. To skip the method, basically we define the metric to find the consistency or similarity of metric tensor from point to point and compare how similar is the effect of the eigenbasis on the other point. What we find is very high correlation across points, around 9.94 across 1,000 points. And in some sense, so this metric tensor or geometry is highly correlated across the space. So the top or non-space will remain the same, which is good to leverage. And there's some global structures in the latent space. Tying this back to the neurons. So we are interested in exploring the tuning functions of neurons on this image manifold. So in a sense, this is defining a function on this manifold, and we should use the geometry to inform our search. For example, if it has a large non-space, we should not search in those space. This motivated our geometric methods for accelerating algorithms. Traditional CMAES algorithm will search Gaussians around each point, which is basically isotropic or spherical. But if you know there's certain directions that change the things faster and some direction that change slower, then you should explore accordingly. That's making larger steps along direction that change slower and to make your sphere more perceptually uniform. So we use this intuition to build the so-called Hessian CMA algorithms, which can search the space using the geometric informations. We benchmark these algorithms on in-silico problems using models as our in-silico units of neuron tunings. And basically, we find we can accelerate or improve our achieved activations in many neurons in using big GAN as generator, and also achieve similar activations, but accelerates the search by reducing the sample complexity in the deep sim generators. And this was used in our future works along these lines, which makes our experiments shorter and monkeys are happier. So as summary, we studied the Riemannian geometry of the generator mappings by computing the Hessians using autograd methods. And we find, despite the covariance was isotropic, the latent space exhibits this highly nonlinear, highly anisotropic but homogeneous geometry. And this knowledge can help us optimize patterns in GANs and interpretation of it. On the hindsight, I find this work exciting because it's a more general framework of understanding nonlinear mappings. We can use it to understand other type of GANs, like VAEs or diffusions, or even the mappings from latent space to neuronal response manifold. And those are left for future work. And that ends the second part. Is there some questions, burning ones? Sorry?
So, okay, sorry. So does that mean you can use smaller or low dimensional, lower dimensional latent space for GAN? Actually, have you tried that?
Yeah, basically that's the case. Like for the GANs we are starting with, the deep sim GANs, it originally have 4,000 dimensions latent space, and in the end we basically reduce it to 500 and have basically the same expressivity. So, and as a coincidence or like correlated fact, after that GANs, most of the GANs in the field are using like 200 or 500 dimensions of the latent space. So in some sense, that may speak to like the true intrinsic dimensionality of the image manifolds, but we have no like clear clues on that yet. Yeah.
I thought the last thing you were showing was super interesting, because when you showed that that null space, at first I thought, oh, that makes sense, because for all the different types of objects, they have their own geometry, like the dog geometry versus facial expression geometry versus forest geometry or something, and you have to pack them all into this space. So to respect all the local geometries, you've got to leave some holes in between them. There's no way it would fit perfectly. So there'd be some null dimensions. Yeah. But then you showed they're super correlated, you know, in different locations in space. So that's suggesting like all those different kinds of objects can be represented with the same kind of dimensions.
Yeah.
That seems really surprising. Like some things like the space of like walking around an object and that kind of rotation, maybe I can understand. But I would have thought that there's other very different dimensions for like human faces versus dogs versus cats. Have you found like the unity of all things?
Yeah, it's very interesting. Also surprising to us, like even you looking at those image manifold, like the big GAN, which is parameterizing or learning from the 1,000 classes in ImageNet. And some classes are like our animals or animate things, and some are like static things, like just a shelf or some things. And it learns the same directions for like changing poses, like doing those things, and like background and smaller, bigger. And I guess that should work for many of the object-ish things. Maybe it doesn't work for some spaces like sceneries or things which are like fundamentally different in some sense. But yeah, comparing across the classes will be quite interesting. And one thing I think underlying this is like for objects, we are all living in like 3D world. So there are certain rules you may obey together, like the latent factors are not really that different. Like there's a 3D geometry and rendering this light and background things. So the real latent factors for this generating process may be similar in the sense so the models happen to align them And maybe for some processes with highly different generating process, like sceneries or like very compositional things, then things could be different, I think. Okay. So that enters the final part of the talk. So which is the alignments of the generative models and the neural tunings. First of all, in the first part, we talk a lot about like the most activating images or the peaks. And if you climb mountain, you know you don't get to the peaks without climbing the hills. So there's a landscape around the peaks, which our generator, our optimizer leverage to climb there. And this landscape is actually the composition of two mappings, which one of them is the generator mappings, which we have just studied, and the other is the more neuroscience classic mappings from the image to the latent space, which image to response space, which is visual processing mapping. So our work has allowed us to study several features on this landscape. For example, we have shown you how to study the preferred images or the peaks. And we have studied like the sharpness of the tunings by measuring the curvatures around these peaks. And we find like there's a progressive sharpening of tunings from V1 to posterior ITs. We can also study like invariances of this landscape, which correspond to the level sets of the mountains. And we find interpretable features of this mountain. Some correspond to things we know, and some correspond to things we haven't known before. We can even find metamers, which can have the same responses, but it means different things because the mountain overlap in a certain ways. But to speak to one question we have raised before, many of these works rely on a single generative models. And what about different generative models? So to motivate that, different generative models can learn distinct latent space and distinct part of the images. And will they be equally easy for neurons to navigate? And there's some patterns of those. So one of the classic questions in neuroscience is about object. And so we tried two types of manifolds. One of them we called it object manifold, which is the big GANs, which I described somehow before. The other one is the deep sim or the pattern-based manifold. So in the big GAN manifold, if you start from the image of the visual and manipulate in the object identity space, it will become a different kind of dogs, rats, or something else. So you change the object identity, but kind of fix the object locations. In the object nuisance transformation space, it will start from the same image, but transform the backgrounds or the aspect ratios of the animals. So in all hand, on this manifold, we have an object on the scenes and keep it in the center of the image. For the other manifold, we call it deep sims. So it's, when you start from the same image of these visuals-like things, it will just put it on different patterns on the canvas without respecting the object structures. So we call it like a pattern-based manifold or more abstract art in a sense. And it has a larger or more expressive space, and it can proxy many images. So in this experiment, we are going to scale up our previous experiments and using like parallel evolutions, which we have two different generators, deep sim and big GANs, running in parallel. And their image generated will be pulled together and form a stream for the monkeys to look at. And the response will be sorted out and feedback to the two optimizers running independently for the two generators. And we have tuned the two optimizers to work optimally for each generator space. The question will be the following. Can we compare the neuron's capabilities through the ventral stream for steering on these two different image manifolds? And the geometry will look like the neuron's tuning function guiding the sampling or optimization of two different image spaces. This is one example you can see. On the left side, it's a pattern-based manifold or deep sim. On the right side is the big GANs. Through evolution processes, the neuron's PSDH, neuron's activation function, will going up. And actually, it's on both sides and will converge to similar activation levels. And even the images looks not like each other's. One is like a crane-like object. The other is like a pattern-based shades. It seems neurons attribute similarly to these two images. And this is a summary of the experiment. of the evolution, one starting from colorless patterns, converting to this, the other starting from like a alpaca-like thing and converts to a crane, but both reaching similar level activities. So giving these results, one thing we can quantify is which space was easier or more facilitating the optimization process. We designed 3 quantification metrics. One is success rate, which is comparing the first 2 blocks versus the next 2 blocks. The other is the achieve maximum activations, which is these values. And final one is the convergence speed, or how fast you converge to the top activations. One thing we find is like the evolution success rate changed systematically along the ventral string hierarchy. So for the deep sim generative models, the success rate in V1 and V4 is approximately 100%, and it drops to like 60% for IT neurons. And that shows like the tuning landscape becomes maybe more complex, or there's more features to climb and it's harder. On the other hand, we find the success rate of the big GAN generator is increasing along the visual hierarchy, starting from around 0% for V1 and to V4's 50% and then around 60% for IT's neurons. So interestingly enough, the rates were also comparable for IT's neurons in guiding the 2-space optimizations. I think the contrasting trend is speaking something. And we find similar results for other metrics. For example, the max activations in V1 and V4, neurons seem to reach higher activation in deep sim space and comparable in IT. And regarding the convergence speed of the neurons, regarding the deep sim, the neurons converge from longer and longer time from V1 to IT. So in a sense, the landscape becomes more high dimensional and you have to climb it using more times. On the other hand, the big GAN's convergence speed is relatively stable and it converges faster in IT than the deep sim image manifold. To summarize, our results are regarding success rate and optimized activations, showing that it's easier or more aligned with the deep sim space in V1 and V4. And in IT, the 2 spaces are comparable. So regarding the evolution speed, in V4, the 2 spaces were comparable, and in IT, it was more efficient in the deep sim space. So there's a decrease of alignment or successfulness going up the visual hierarchy for deep sim, and the opposite trends for big GANs. You may ask, like, how should we interpret such results? One of the more hand-wavy interpretation is that maybe there's a certain kind of language they speak that are different from each other. For example, if the big GAN generator creates a cat of some sorts, and the V1 neurons of the classic ones said yes because there's the Gabor patches on this cat's head, then the optimizer will get a wrong message and create more kind of cats for it, and some will be tabby ones, some will be not. So the neurons will be confused and not going to optimize this space very well. So I think the intuition is that the tuning axis of the neurons and the parameterizations of the GANs should be somehow aligned to each other to maximize the successfulness of the evolution experiment. And one thing we know for neurons is that they do not just give us scalar numbers, but they give us the dynamic traces of the PSDH. So how does that interact with the evolutions? So what we find is that before the optimization procedures, they can generally evoke a higher PSDH throughout the time trace. However, after a optimization profile, PSDH seem to have dynamic preference in IT, where the earlier part will prefer to the deep sim or the pattern-based manifold, the later part will be preferred to the big GAN object-based manifolds. While in V1, V4, there's no such dynamic switch of the preference. We quantify this effect by attributing which part did the neuron recruit in their optimization procedure. So the objective is set to optimize the average firing rate in the 50 to 200 milliseconds. So neurons, in some sense, could free to choose which part they are optimizing or increase their firing rate. And Some neurons can increase their transient activity, some can improve their tonic activities. So we can attribute which part does the neuron recruit. What we find is that there's no significant difference in V-force for their improvement in two spaces, but for IT, there's a preferred recruitment of early response for deep sim and the preferred recruitment of later response for big GANs. So it seems like the optimization facility or easiness for two spaces are also time segregated, and there's a dynamic preference of two spaces. So previously, we find IT neuron seems to be ambivalent to the two spaces and aligned similarly to the two generator. But if we look more closely about the temporal responses in early one and later ones, seems like the early one is more preferred to like deep sim, just like before, and later ones are more similar to the big GAN object manifold. So maybe the recurrent processes of the visual cortex may be implicated in this preference. Finally, how to interpret this result and what's the theoretical framework to understand this alignment thing? As we have showed, the optimizer will operate on this composition of mappings of tuning landscape. And so the hypothesis is, if you want to steer the latent codes, this landscape should be relatively simple, maybe linear or like Gaussians or like a single bump. Or it could be non-optimizable because it's too nonlinear. So we did a toy examples showing this effect. If you basically have a Gaussian tuning functions and then distort it using invertible maps or real NVP, you will distort the landscape to be more and more complex if you go through more and more layers of these networks. What you find is that if you go through more layers of the nonlinear transformations, even though there's still unique peaks on this landscape, it's becoming hard or even impossible to find it using the algorithm we use for neurons or CMAES algorithms. And this effect is more prominent for deeper networks like higher dimensional spaces like 256 dimension spaces. So in some sense, we can infer this alignment through the easiness optimization. If some space or composition of mappings is easier to optimize, maybe there's a simpler tuning landscape of the composition of the two functions, and maybe these two functions are better inverse to each other's, like the GF is able to invert the effect of G, so the mappings, it becomes more linear. So we did this computational simulations of this effect. We used the two generators, DeepSim and BigGAN, as our image generators, and used in silico units from CafeNet as our neural tuning functions. What we find is that for BigGANs, if you compose these two mappings and do linear regressions from latent codes to neural responses, you'll be better and better at predicting the response if you're going deeper for the visual hierarchy. So the higher visual cortex is more linear to the latent codes in the generator space. For DeepSim generator, although it's more high dimensional, it did a poorer job for predicting higher level visual cortex responses in silicos. And also the trend is similar to what we see in vivo, which is there's an increasing trend of predicting vector more linearly to higher visual cortex responses in big GANs, but there was a decreasing trend in deep sims, or better, worse and worse. So in summary, maybe we can think about visual system as an inverse to of the generative models of the world. So from the pattern-based manifolds, there's a decreasing of alignment, and the object manifold, there's an increasing alignment, and even better for later responses in the neurons. And so the hypothesis or theory may be the sensory system learned to infer the important latent factor or latent codes using in some of the generative model of the data distributions. So summarizing this part, we find going up the ventral hierarchy, neuron tunings on the object manifolds became easier to climb, and the posterior IT neurons has a comparable success rate in both spaces. And dynamically, later response of posterior IT was more aligned or optimized easier on the object manifolds, and the early response is more to the pattern manifolds. And so maybe the theory to explain it is the visual hierarchy provides a way to better infer the inverse the object manifold generative functions. To summarize this talk, in the first part, I told you one approaches we built to use neuronal feedbacks to do closed-loop visual neuroscience experiment and explore the visual world in a more automated way. In the second part, I told you how we understand the geometry of generative models. And the third part, I told you how the neuronal tunings aligns with this latent hold geometries. So moving forward, there's some very immediate extensions of our work. Maybe we can extend our works beyond visions. So we can do some changes to the search domains, for example, for auditory space, for vector space, or even neuronal stimulation space. Or the search objectives can also be different, like population responses to steer the population vectors or reconstruct it, which are some directions we are currently doing. And you can even steer like perceptual behaviors or using different modality of data like fMRI or two-photon imaging data. From the third part, also we have just scratched the surface of generative models. We only use two types of models from like 7 years ago. And there are so many kinds of new generative models which learn from not just image world, but the world of dynamics, of videos, and other type of data. Perhaps they will learn some interesting generative factors of the more broad sensory world, and then they will be related to the neurons in certain ways. Finally, what motivates my research is this study of creativity and imaginations. And some models, like the generative model themselves, has become interesting models of creativities. So can we study these models and understand how they are creative, like how they decide what to paint in their painting process? And maybe that will be biological plausible or relevant to our imagination process, like how to implement those models in our brains and implement our imagination process. So in the past, I've shown you my past work in closed-loop visual neuroscience, and in the future, it could become closed-loop sensory neuroscience, or measuring the general alignment between brains and generative models. And I have done some work for understanding generative models themselves. And in the future, I'm planning to adapt these models as a part of the model of our generative process internal to us. And hopefully, this line of work will bring us towards the generative neuroscience futures. And this is the acknowledgement, and thank you for your attention.
All right, very good. We have time for a couple of questions.
Hey, Pinshu, that was a really nice talk. Hi, also good to see you again.
Yeah.
For the neural experiments, you guys were all doing it in awake monkeys, right?
Yes.
Do you think that any results would be significantly different if they were done in anesthetized?
I think it will be even better for anesthetized. So I think there are some classic work saying that the anesthetized responses is like, in some sense, weaker, but less noisy or less variable. So it's in some sense more like a static function. So I imagine it will be even more optimizable in the sense.
Yeah, so I mean, also kind of going off of that, like I'm not familiar enough with these GANs to have a sense, but like if feedback is usually baked into these things, but I was wondering if you have some sense or like speculation about how feedback across different levels might change these kinds of like manifolds.
Very good point. Yeah, so through the talk, I have been pretending that this dynamic, I mean, there's more or less dynamics in this function. So we are pretending there's a single function mapping the images to the responses and it's static across time. And we know it's not the case. And so definitely in the last part, you have seen like the tuning functions of IT neurons showing this dynamic switches from the more V4 early, like the pattern preferring to the later ones. And we guess there will be some effects of like recurrent or even feedbacks from a higher visual hierarchy. If we are having to record from there, maybe we can find correlations between these two spaces.
There's a question on Zoom. You may be able to read it on the podium monitor.
It's from David Van Essen. So he asked, like, were most of your recordings from multiple units rather than single unit? If so, how does that impact your interpretations? Very good questions. So because it's like a multi-electrode array recordings, we have less control of what kind of unit we record from. So we got single unit and multi-unit simultaneously, and single units is like more like come and go-ish. So in our first paper, we systematically compared the two units, the single unit and the multi-units recording in the same electrode. Their tuning functions are highly correlated. So basically, if you manipulate the image around the image manifold, the mountain will look very similar to each other's. And also the images they create will be similar. So let me see. The major difference we find in single and multi-unit in that case is that the single units will have a sharper tuning peak, in the sense it's more selective, and multi-unit is broader tuning peak, so it's less selective. Perhaps because averaging multiple single units of the similar tunings, because maybe like the column structure in visual cortex. Is there an example of such? Oh, there's a. Oh yeah, so it's not the best examples, but if you measure like we call it like tuning maps. If you manipulate the image on the 2D grid around its peak, then you see this kind of 2D tuning function along some submanifolds. And what you see for multi-units, like it's broader and extends towards like the boundaries, but a single unit will be more like the centered things, perhaps because of the flowering effect or like the more sparse activities. Yeah.
The questions.
Hi, Vinci. Yeah, thank you. That's amazing work. So my question is more about so the last slide summary you showed the differences between these two. But I always really wonder when you try to predict IT, basically there's only 2/3 of them can get successful. So my question is more about, okay, so what is missing there, and do you need another model? From your future work, you kind of say you may want to actually use some auditory input and other things, but I think one of the things, if from artificial intelligence point of view, very important is not, I mean, the visual stream, I think it's not just try to see stuff, right? It's not just like, okay, I understand, I recognize. The more important is the visual computation. And I think people here know that, right? You can, when you use a DALL-E or other kind of assistant, it's actually, they are very bad at using visual computation. Like you do the works with how to manipulate things in a visual sense. So I was wondered, I think that's the frontier of artificial intelligence and how you think you're going to go forward, or if that's a plan. Thank you.
Yeah. Super good questions, and I think most of us, if you use DALL-E or some sort of artificial generative models, you find like some frustrations in like manipulating or prompting them in a certain ways. And especially failure case is kind of this spatial kind of relationship case, like how they interpret one thing is on top of each other and on side of each other. Many of them still don't do it correctly currently, and we have some work trying to study why they fail in some sense. But indeed, like hopefully, if they learn from the right data distributions, they can learn to interpret this relation in the correct ways. And currently, all our generative models are basically in like single object image data, like ImageNet. So there's basically like no sense of relationship of this kind of sorts. So if we learn from the broader distribution of the sceneries or compositional things, perhaps it will learn something like this relationship, which may be related to our processings, like in the visual computation sense.
Thanks for the talk. I'm thinking that one of the goals of neuroscience is to understand behavior by knowing something about the network.
Yeah.
So I'm not quite sure that I can extract that sort of information from your work. So other than that, there is a hierarchy,
yeah,
and there are recurrent networks.
Yeah.
And that's it. Or is there more to it, which you can sort of pinpoint exactly what we should be looking for when you're trying to understand the structure of the brain?
Yeah, very good point. Like in the talk I've gave today, I have basically no behavioral components. In some other work that done alongside with me, people show that the images they extract from posterior ITs, they have behavior effects. They elicit longer fixation time. If monkey are freely viewing, they will prefer viewing some of the images. And it's from posterior IT, so it's not on the top of the hierarchy. So perhaps if you record from the more anterior regions and evolve certain things, they will get the visual features that are more relevant to behavior. For example, it's like the salient things you see in your visual world and will attract your attention or attracts your fixations. And those things may be relevant for downstream learnings and other things. But currently, I didn't show you those results.
I'll take the host prerogative and ask one more, and then maybe we should close it there to give you some time to set up for your next thing. So I guess from some of your work and especially even in some of the sort of model artificial systems you set up, I'm wondering if you can comment on where you think we are in this sort of overall program. So here's where I imagine it is. And let me give an analogy to other sort of optimization experiences I've had. On many problems and with many algorithms, when you try to optimize something, you often find that there are two phases. One, there's a rapid descent phase, and you can quit there, or you can keep going for a long time, and it takes forever, but you might end up at a very different place. The analogy I'd sort of give is if you got a pair of skis and you're wealthy enough to have a helicopter, and you jump out of the helicopter at some random point, you're likely to end up very high on a mountain. You're going to lose altitude very quickly at the beginning until you get to a valley. From that point on, you're not going to change your elevation that much, but you're going to end up in a very different place from where you started if you follow the valley down to sort of base camp. And that sort of horizontal travel, I imagine, being like the character of the images that you're discovering. And the vertical altitude is like the firing rate of the neuron. So you start out with bad firing rates, you very quickly improve those, but what is the likelihood that we're really sort of discovering stimuli that are anywhere really even close to, sort of optimized for the neurons is the sort of nature of the question.
Yeah, that's a very good question. I think one of the plot basically speaking to your comments, the first animations I showed for evolution basically have that two phases. You can see it, this one. So there's indeed so indeed at first there was very fast increase of firing rate, and then things start to stabilize, and there's a prominent feature, but things start to be slower optimizing, and firing rate start to increase lower. And in some sense, the second part is also interesting to us because it's kind of walking in these valleys. And so in some sense, that is the invariance manifold people are looking for. So if neurons encounter stimuli in that regime, then it will be relatively similar activations. So one thing you can do is to maybe do PCA or do some sort of analysis of the latest regime, and then they will reveals what type of things neurons are less sensitive to. And that's also interesting. Yeah.
Very good. Okay, thank you very much. Let's all thank Bing Xu again for a great talk.